Okay, so this is the first embryology video that I'm going to be doing for the first year medical students, and it's going to start at the point where a sperm has met an egg and a zygote has formed, so you now have a diploid cell. The diploid cell begins to rapidly reproduce, and you end up having a cell that looks roughly like this. It's called a morula, and it basically looks like a blackberry. It's lots and lots of cells bundled together, and it starts becoming called a morula when it gets to about 16 cells. This continues to proliferate, but steadily the zona pellucida, which still exists around it, begins to break down. As it breaks down, you end up with a cell that looks something like this. This is called a blastocyst. The blastocyst is the name of this cell at the point where you have an outer layer of cells called the trophoblast layer, which runs around the outside, and an inner layer of cells called the inner cell mass, which is just there. So at this point there are only two separate types of cells, your trophoblast cells and your inner cells. This then continues to proliferate, only because the zona pellucida has disappeared, it is now more and more easy for fluid to enter the cell, and so you end up getting the formation of a second pocket. So underneath the first one, you will have noticed a gap here, which was just called the blastocyst cavity. Now, with the formation of a second cavity here, it becomes known as the yolk sac, and the second cavity becomes known as the amniotic cavity. You have two layers of cells that you can just about see, an upper layer called the epiblast layer and a lower layer called the hypoblast layer, here and here. So this is the first point at which you start developing what you refer to as the axes of the fetus. From this point onwards, the layer that is represented by the epiblast is the more posterior element of the fetus, and the hypoblast layer is the more anterior element, so back and front. At this point, you should have started to have something called implantation. This is where the fetus, or zygote, begins to embed into the maternal endometrium. As it starts to do this, the trophoblast layer that we talked about earlier starts to invade into the endometrial tissue of the mother. So your cell, we've been talking about already, lies roughly here on a diagram, and your trophoblast layer, which had been running around the outside, has now differentiated into two separate layers. You've got an external layer here, called the syncytia trophoblast layer, and an internal layer, which is only 1 or 2 cells thick, that still runs around the edge of what once would have been the trophoblasts. The difference between the two is indicated by the syn bit in the name of the syncytia trophoblast. Syn, it's coming from words like synthesis, is because the cell membranes of your trophoblast layer have broken down and you end up with a polynucleated cell mass. This is what's secreting all the hormones and proteins necessary not only for the breakdown of the maternal uterine lining, but they also produce really important hormones such as HCG, which is necessary for the preservation of the corpus luteum, which is still producing the estrogen and progesterone necessary for the maintenance of the uterine lining to prevent breakdown. But at this point, the fetus won't have fully embedded into the uterine lining, and so you still have an area of exposed uterine cavity. Okay, so at this point, the cells keep proliferating, and you've got two separate layers. The next thing that happens is you develop a third layer, but to develop a third layer, firstly, you need to have this form. It's called a neural groove, and it's basically a small dip that forms in the middle of the epiblast layer. And what happens from here is you start to get cells migrating along this line here, coming in through here, called invagination, and they then displace first the hypoblast. So you get a line of what was once epiblast layer cells there. And then you get a second layer of cells coming through here, forming a middle layer, and then they displace their own cells here. So you end up with 3 separate layers of cells, all derived initially from what was once the epiblast. This is called the trilaminar disc, and occurs while the fetus is still continuing to implant into the uterus, And so the external layer should look something like this at this point. So you have a continuous layer of trophoblasts and syncytiotrophoblasts surrounding an inner cell where you've got two layers, epiblast, hyperblast, that have now become three. 1, 2, and 3. So those three layers are going to be called completely new things from now on. So you've got your first layer, which previously was called the epiblast layer, is now called the ectoderm. The middle layer is called the mesoderm, and the bottom layer is called the endoderm. So ectoderm, mesoderm, and endoderm. So this is called, as I said, the trilaminar disc, and is a continuation of the formation that we talked about a minute ago. The one other thing you might notice on this diagram is you've got a little bundle here, right at the bottom, of cells that have aggregated together and become known as the notochord. Now, the notochord is really important because it is responsible for the development of something called the neural tube, and the neural tube forms at a later stage forms the spinal cord of the fetus. So as I was saying earlier, the outer layer of trophoblast has been continuing to develop, and you might have noticed there are these little pockets around the outside. So these pockets are just gaps within the syncytiotrophoblast, as the syncytiotrophoblast has been growing outwards and outwards and outwards. Eventually they'll become filled with the maternal blood, and as some of you might have guessed at this point, this is the beginnings of a really early placenta. When we talked about that mesoderm forming in the middle, when it came through the neural groove and moved outwards laterally, it began to move around the outside of the two other layers and completely enveloped them. And so if you were to look just at that central layer from a distance, it would look something like this. Your ectoderm and your endoderm with mesoderm completely surrounding it. So while there is a trilaminar disc still in the centre, in other places it appears as if you have 4 layers. At that point, your mesoderm is split into 2 parts, the superior and the posterior and anterior lateral plates of mesoderm. So the next thing that happens is linked to that notochord which we talked about a minute ago. So your notochord at this point stimulates something called neurulation. Now, neurulation is the formation of the neural tube and is absolutely vital for the fetus because the neural tube becomes a nervous system for the fetus, and without a nervous system you won't get proper sensory input. So the notochord stimulates this process. It's called budding, and the ectoderm, 2 edges of the ectoderm rise up, pinch together, and eventually these cells will join in the middle and form a complete tube, and these will join and you'll form a layer. So you'll end up with your 2 plates of lateral mesoderm, a bit of intermediate mesoderm, a notochord, and some endoderm. Now some of you might have noticed that I've drawn a little line of cells running from here all the way outwards. Now this is because that lateral plate mesoderm that was out here, when it first came out, was one continuous cell mass. Slowly ends up filling with fluid in the same way that the morula began filling with the fluid to form a blastocyst, and then continue to fill with fluid to form an amniotic cavity and a yolk sac. Here you get what's called an extra embryonic coelom, and again it's filled with fluid and completely surrounds the fetus, separating it from your outer layers of trophoblast, syncytiotrophoblast with cytotrophoblast. But a small layer of that mesoderm that migrated outwards lines the inside of your cytotrophoblast, and this combination here forms what's called the chorionic villi. Now the chorionic villi are really important because this is essentially the miniature unit within the placenta and creates a surface area that's vital for gas transport. So once you've got the formation of your neural tube and your chorionic villi begin to form, you end up getting something that looks like this. So you've got a neural tube in the middle with ectoderm above it, intermediate mesoderm, lateral plates mesoderm and endoderm, with an amniotic cavity above and a yolk sac below. And again, you've got a gap of extra embryonic coelom with mesoderm, cytotrophoblast, and syncytiotrophoblast, which is that polynucleated cell mass that has broken down membranes within it. And if you look at this bit from a slightly more zoomed out and simplified perspective, it should look something like this. So you've still got that lateral plate mesoderm wrapping around it, and you would have another layer of lateral plate, and you would have another bit of mesoderm roughly here, slightly further out. These small diagrams that I'm showing from time to time on the side are basically a zoomed out, simplified version of what's happening internally to the fetus itself. My outer circle here is the placental development. So when you're going through this later or in your own time, you can think about it in completely different layers, just from a cell centre perspective, from a zoomed out bit of the embryo, or joining everything together with the placenta. Moving on, you end up getting differentiation of that intermediate mesoderm in the middle. So your intermediate mesoderm, which lies roughly here, ends up getting two bits of it differentiating into a slightly different type. The most medial, so the most central bit of intermediate mesoderm, becomes called a somite. And then the next bit along becomes called, well, remains called the intermediate mesoderm, separating the somite from the lateral plate. These could take on completely different roles later, and in a second this somite from each side will join together and envelop the notochord and the neural tube, forming a ring around them. They'll be important for nerve development and for muscle and bone development later. This little bundle of intermediate mesoderm that's slightly separate to the lateral plate mesoderm is going to be important in gonadal and kidney development. So as you can see, you've had those somites, those inner layers of intermediate mesoderm have now wrapped around your neural tube and notochord and formed a bundle, which will later form the sclerotome, dermatome and myotome layers. Anything that I refer to in this video, where the names are unclear or uncertain, they're all covered in the notes that come attached to the video, and so look at them for reference if you're unsure about anything or spelling of names. But at this point, you've got some very important layers. You've got your ectoderm layer, you've got your mesoderm layer, your endoderm layer, with your primitive gonads and kidneys, your nervous tracts surrounding your neural tube and your notochord, and these plates of lateral mesoderm over here. So now I'm going to backtrack a little bit. I'm going to backtrack to the point where you first started talking about the trilaminar disc. So when you first started talking about the trilaminar disc around here, you started to have a process called gastrulation occurring. Now gastrulation is really important because it's what creates all your internal organs, certainly the vast majority of them. But to understand it with all the things associated with the neural tube involved gets really complicated. So I'm going to simplify it all by stepping it straight back to a point where all of this looks simply like three layers. Now these three layers undergo a folding very, very similar to the folding that happened with the neural tube development. So firstly, they sort of bud downwards. So whereas the neural tube budded round, these ones bud downwards, and they begin to join in the middle. That joining, as I'm sure you can see, forms something like this. So you've got your 1, 2, 3 layers of your trilaminar disc here, but also 1, 2, 3 layers of trilaminar disc at the bottom, and then 1, 2, 3 layers of trilaminar disc here. And that looks really complicated, and I'm sorry, it's not particularly easy, but that happens for all embryos at this point, and is what forms your GI tract, which essentially ends up becoming just here, this most central ring there. So looking at that from a slightly zoomed out perspective, you've got your 3 layers here, but your mesoderm, which we've already said was split into lateral plates, still has to envelop absolutely everything and absolutely everything here. And then you've got your endoderm at the bottom. So you end up with endoderm, mesoderm, ectoderm, and then you have your whole separate bundle here. If you need to look through that again, keep going back over the video because it's quite a complicated process. But what you end up with, if you can imagine your embryo used to look like this, if you go back to a slightly more complicated image, your embryo used to look like this, with your ectoderm, neural tube, notochord, mesoderm and endoderm. That folding round causes it to become something slightly more like this. So your neural tube ends up here, your notochord, your somites have wrapped around it, your lateral plate mesoderm comes and bananas around because this joins round here, and you've got a small bit of primitive gut that's trapped in the middle, and this forms your GI tract from top to toe, and then you've still got your yolk sac down here and amniotic cavity around here. Okay, and so broadly speaking, that is all the embryology up to the point where you now have a placenta with your chorionic villi here, a gap between your placenta and the fetus called your extraembryonic coelom here, and then within your fetal area you've got a yolk sac, an amniotic cavity, a neural tube with notochord, a primitive gut, and your lateral plates of mesoderm with your kidneys and primitive gonads directly above it. Hopefully that's everything, and as I've said already, everything here is typed up in notes to accompany the video, and if you go forward and backward and think about taking the layers on and off, so having simpler images and then making them more complicated with placenta, hopefully that's everything.